Welcome back to this next video in which we are discussing the thyroid gland. Okay, so in this next video we want to discuss what the effects on peripheral tissues of the body that T3 and T4 are going to have. Okay, so before we actually say anything further, let me just remind you of the fact that T3 and T4 levels within the blood do not really fluctuate physiologically. They are kept at a constant level by the thyroid gland. So the thyroid gland matches the amount of T3 and T4 that it's putting into the blood to how much actually needs to be put in order to maintain a constant concentration of T3 and T4 in the blood. Okay, so remember, these are not hormones whose level is fluctuating. It's not like, for instance, adrenaline, where you get a huge rise in the adrenaline level in the bloodstream during a stressful situation, and that brings about the fight or flight response. It's not like that. Whatever T3 and T4 are doing, they're doing it all the time, and when we discuss these effects of T3 and T4, it's worth remembering that. Okay, so let me now talk about the actual peripheral effects of T3 and T4. So I'm going to talk about three major effects of T3 and T4, and I'm going to, as I say, split this into three categories. So the first category of effects of T3 and T4 is in a tonic mobilization of energy sources into the bloodstream. So for instance, they're going to activate the liver to be putting glucose into the bloodstream. They're going to be activating the adipose tissue to be putting free fatty acids into the bloodstream. They're going to be activating the skeletal muscle cells to be putting amino acids into the bloodstream at a tonic level. That's the first effect that I'm going to discuss, and I'll just list them off firstly and then go through them all in more detail. Secondly, they're also going to help maintain a constant rate of heat production by the body. Okay, thirdly, they're also going to help to maintain sympathetic nervous system tone to the heart. So they're going to be involved in maintaining normal heart rate and normal heart contractility. Okay, so those are the three categories then of effects of the thyroid hormones that I'm going to go through. So we'll begin by discussing the mobilization of energy sources into the bloodstream in more detail. Right, so let me draw pictures then. So we're going to have effects, as I say, on the hepatocytes of the liver, on adipocytes in the adipose tissue, and on skeletal muscle cells. So I'll start by drawing the liver here. Okay, so here is a basic picture of the liver, and then we'll have a hepatocyte within the liver shown a little bit out of scale here. Okay, so T3 and T4 then, and I'll actually just put T3 and T4 right at the center of this picture. So I'll just put T3/T4 to mean either of them, because we know that they both have the same effect, although T3 is slightly more potent. They're going to go into the hepatocytes, and they're going to activate a tonic level of hepatocyte release of glucose into the bloodstream. And the way in which they're going to be doing this is they're going to continuously be having a pro-glycogenolytic effect on the hepatocytes and a pro-gluconeogenic effect on the hepatocytes. So they are going to be continuously activating both the process of glycogenolysis, which means the breakdown of glycogen. And remember, glycogen is a polymer of loads of glucose molecules that is stored in hepatocytes. So glycogenolysis means the breakdown of glycogen polymers into glucose monomers, and then those glucose monomers can be released into the bloodstream. So glycogenolysis is one of the things they're going to be activating, and they're also going to be activating the gluconeogenesis process. Now let me just remind you of what is meant by gluconeogenesis. Gluconeogenesis is the synthesis of glucose from amino acids. So you start with amino acids, the constituents of proteins, and you can convert those amino acids into glucose. So this means that the hepatocytes are going to be needing to receive a supply of amino acids in order to actually be participating in gluconeogenesis, and we'll see that T3 and T4 also activate skeletal muscle cells to continuously be releasing amino acids. Therefore, those amino acids that the skeletal muscle cells are releasing are what the hepatocytes will actually be using for the gluconeogenesis process. So I'll just put a little bit more detail on here. So glycogenolysis then is the breakdown of the glycogen polymers stored in the hepatocytes to release glucose monomers, whereas gluconeogenesis is the conversion of non-carbohydrate molecules, specifically amino acids, into glucose. So there's a little bit of a summary of what these two processes involve. So T3 and T4 then, the centres of all of this, they are tonically activating hepatocytes to release glucose from glycogen stores, and also to be converting amino acids which they can receive from the blood from skeletal muscle cells into glucose. Okay, so the mobilization of glucose into the bloodstream then is one of the things that T3 and T4 are continuously activating to some degree. Now, I know what you might be thinking at the moment. Hang on, but, you know, insulin and glucagon control this, don't they? Not T3 and T4. I will put this all in context in a moment. This is sort of like a background effect happening all the time, and then on top of that you're going to have insulin and glucagon, which of course are these hormones that are majorly involved in controlling metabolic processes in the entire body and switching us between a state where we're in the fasting state and we need to be putting energy sources into the bloodstream, and when we're in the fed state where we need to be taking energy sources out of the bloodstream. But I'll put this all in context with insulin and glucagon later on, so I'll put insulin and glucagon on top of this in just a moment. Okay, but for now, understand that T3 and T4, we've got this sort of tonic level of them within the bloodstream, and they're going to be activating hepatocytes always to be releasing glucose into the bloodstream. And where are these hepatocytes getting that glucose from? Well, from the glycogenolysis process and also the gluconeogenesis process. Okay, so now let's talk about the effects of T3 and T4 on adipocytes and also on the skeletal muscle cells. So I'll start with adipocytes. So I'll draw a big white adipocyte here, and I'm going to now specifically stress the fact that it is a white adipocyte, because we will also be mentioning brown adipocytes. Now, of course, in the adult human we don't think we have many brown adipocytes, so often we just refer to white adipocytes as just adipocytes because there aren't any other types of adipocytes. However, we're going to see that T3 and T4 in the neonate, the human neonate, can have effects on the brown adipocytes. So because we're going to be talking about these two different types of adipocytes, I am now stressing the fact that this one is a white adipocyte, and I'll refer to it as a WAT, so this is white adipose tissue. Okay, so of course white adipocytes, they have this structure where they have a massive great droplet in the centre, a massive great fat droplet here, which I'll colour in yellow here, and that's where loads of triglyceride molecules are stored. So white adipocytes, they have a great big fat droplet in the centre of them where they're storing loads of triacylglycerols, or triglycerides for short, so there's lots of tag molecules in here. Now, T3 and T4 molecules will continuously be having an effect on these white adipocytes, and they're continuously going to be activating lipolysis process. So they're going to be activating in these white adipocytes the lipolysis process. And in lipolysis, what's going to happen is the triacylglycerols are going to be broken up into a glycerol molecule and a free fatty acid molecules. So if I just draw this, so here is a little cartoon of a triacylglycerol molecule. So this is the glycerol molecule here, and these are the free fatty acids attached onto here. Okay, so this molecule here is representing the glycerol backbone of the triacylglycerol, which I'll underline now in green, and these orange horizontal lines here, these are representing the fatty acids. And remember, fatty acids is just another name for long chain carboxylic acids. These structures are really just incredibly long carboxylic acids which have been esterified onto the alcohol groups of the glycerol molecule. Okay, so in lipolysis then, what's going to happen is these triacylglycerol molecules, which we have stored in the fat droplet of the white adipocytes, are going to be broken down. So we're going to hydrolyze the ester links here, here, and here to break this into a glycerol molecule and free fatty acid molecules. So first free is the number free, and then the second free is free as in not bound to anything. So free fatty acid molecules. Okay, and then these fatty acid molecules, they can be released into the bloodstream and they'll bind to albumin proteins, and they can be delivered to tissues such as the heart, which can use them in the beta oxidation process to derive energy from them. Okay, so T3 and T4 then are going to result in free fatty acid level in the bloodstream maintained at a tonic level. So they're going to be activating tonically the release of free fatty acids from the adipocytes into the bloodstream. Okay, so I'll just put this here. So the things that these are going to be putting out are free fatty acids, which are often abbreviated down to FFAs. Okay, so the adipocytes are going to be tonically releasing free fatty acids into the bloodstream under the influence of T3 and T4. Then finally, T3 and T4 also have an effect on the skeletal muscle tissue, so I'll just draw a skeletal muscle here. Okay, like so, and here this can be the tendons. So I'll colour it in red to make it look more convincing. And what do skeletal muscle cells, what's the major thing that they store? Well, the major thing that they store is proteins. They have a huge number of proteins in here. So what they can do tonically under the stimulation of T3 and T4 is break down some of these proteins, a process known as proteolysis, okay, and then release amino acids into the bloodstream from these. So I'll just, because of the lack of space, I'll abbreviate amino acids down to alpha-alphas, so this is often an abbreviation for amino acids. So T3 and T4 can they do trigger a tonic level of proteolysis by the skeletal muscle tissues, so that you're getting amino acids being released into the bloodstream to keep amino acid level the same within the bloodstream. Okay, right, and these amino acids can go off to the liver cells, the hepatocytes, and be used in the gluconeogenesis process. And of course they can be used for other things around the body as well. Okay, so this is what T3 and T4 do. This is one of the major effects of them. So this is category number 1 of the effects of T3 and T4, that they continuously, tonically trigger mobilization of these energy sources, so carbohydrates, lipids, and proteins here into the bloodstream. Okay, now, what I want to do is just put on top of this a reminder of insulin and glucagon, because insulin and glucagon are the ones that you will be more familiar with in controlling these processes rather than T3 and T4. T3 and T4, remember, they're always there and they're always doing this. So this is, if you like, a process that is occurring in the background to glucagon and insulin. Glucagon and insulin are the dramatic ones that hugely change their effects depending on whether you're in the fasting state or the fed state. Whereas T3 and T4 are just always there in the background, activating the release of molecules into the bloodstream for energy. Okay, so let's just have a little reminder of insulin and glucagon. So remember, insulin and glucagon, it's like a seesaw, it's one or the other basically. So you switch between an insulin dominance in the fed state and a glucagon dominance in the fasting state. Okay, so in the fed state then, when you've just eaten a meal and you've got loads of glucose molecules coming into the bloodstream from the gastrointestinal tract, you've got loads of amino acids coming into the bloodstream from the gastrointestinal tract, you've got loads of lipid molecules coming into the bloodstream from the gastrointestinal tract, insulin dominates. You have lots of insulin in the bloodstream and very little glucagon in the bloodstream. And when insulin is high, insulin is the signal to all of the cells of the body that it's a time for a feast, okay? People often say when they're asked what is the function of insulin, the quote that people generally give is it's to lower blood glucose. That is not incorrect, and it is what a lot of people will say. However, you've missed the point of really what insulin is all about. Insulin is this signal that goes up in the bloodstream and it tells all the cells of the body that we are in the fed state. We have a huge amount of food available, and it basically tells cells all over the body that now is the time for them to feast. It gives them the permission to take loads of energy molecules out of the bloodstream and use them for their own purposes. That's what insulin is doing. So, for instance, it tells the hepatocytes that now is the time for them to take glucose out of the bloodstream rather than put the glucose in. So they can take glucose out and store it in glycogen molecules. It tells the skeletal muscle cells to stop breaking down proteins and releasing amino acids into the bloodstream, and instead start taking amino acids and glucose up into themselves so that they can start rebuilding proteins. Okay, it tells the adipocytes that now is the time not to be breaking down triacylglycerols and releasing free fatty acids into the bloodstream, but instead let's take up glucose and other molecules and turn them into fat molecules. So it causes all the reverse of what's happening with T3 and T4, but T3 and T4 will always still be there, having an influence, saying the opposite to insulin. But of course insulin dominates in this fed state, so the effect of T3 and T4 is just to sort of keep insulin a little bit under control, but it's not going to actually reverse the effects of insulin. So you just have to remember that this is in the background to insulin, and insulin will dominate when it's there. Glucagon, meanwhile, is the one that goes up during the fasting state, and this really just has an effect on the hepatocytes. So in the fasting state, insulin goes down. Remember, it doesn't go to 0, but it goes down a lot, and glucagon goes up, and glucagon is a signal that tells the hepatocytes that they need to be releasing glucose into the bloodstream rather than taking glucose out of the bloodstream. So it works along with T3 and T4, and also along with the low insulin levels. So the low insulin levels mean that we're now going to have low signaling telling the hepatocytes to actually be taking glucose up, because remember, insulin is the signal telling hepatocytes to store glucose, take glucose out of the bloodstream. So low insulin means that they're going to be not doing much of that, and then high glucagon means that they're going to be doing the opposite. They're going to be activating glycogenolysis, the breakdown of glycogen to release glucose, and also gluconeogenesis, the conversion of amino acids into glucose. So glucagon will be majorly activating the release of glucose into the bloodstream, but of course T3 and T4 will always be there in the background helping glucagon along. Okay, glucagon does not really have an effect on white adipose tissue or the skeletal muscle. Instead, it's the low insulin that has an effect on the skeletal muscle cells and the adipocytes. So low insulin means that the skeletal muscle cells are going to not be taking glucose and amino acids out of the bloodstream and storing them as proteins. Instead, they're going to be breaking down proteins and releasing the amino acids into the bloodstream, and of course T3 and T4 will be there in the background helping out with that. And also low insulin is going to be causing the white adipocytes to move over to now breaking down triacylglycerols and releasing free fatty acids into the bloodstream, and of course T3 and T4 will be in the background helping out with that. So really what that discussion was aiming to do was to make you understand that T3 and T4 driving this release of these different energy storage molecules into the bloodstream is in the background to the glucagon insulin system. This dominates, okay? When insulin goes up, it dominates over this. This becomes irrelevant. Insulin is in control during the fed state. But remember that T3 and T4 will always be there in the background, reducing the effect of insulin slightly. So if it was the case that T3 and T4 were taken away, you'd see exaggerated effects of insulin because now they weren't being reduced to that little bit by the T3 and T4 signaling. Okay, but remember that this dominates over this during the fed state. So that's on top of this, if you like. Okay, so that's what T3 and T4 are doing with regards to mobilizing energy sources into the bloodstream, and they're doing that all the time. Right, so now that we've been through that, let's go on to the second category of effects of T3 and T4, which is all about maintaining body heat production. Now, there's two different parts to this. Firstly, I want to talk about what is understood about how T3 and T4 maintain body heat production all the time in adult humans, and then I'll talk about what we know about how it helps in the human neonate, and also in other mammals, if you care about other mammals. Other mammals do have brown adipocytes in the adults, and they're involved in thermogenesis in those other species, but in humans we don't think that adults have brown adipocytes, so it's only relevant for human neonates. Okay, right, so firstly let me give you the pathway that we do know is present within adult humans for producing heat, and this is not involving brown adipocytes. Okay, so I'll just put the title here. So this is heat production, and of course I'll remind you again that T3 and T4, they're at this constant level within the bloodstream, and therefore they're continuously going to be activating this heat production. So they activate a tonic heat production within the human body, and if you were to have too much T3 and T4, you'd get overproduction of heat, so you'd be feeling hot all the time, and if you had too little T3 and T4, you'd get underproduction of heat, so you'd be feeling very cold all the time. Okay, so heat production, firstly, what we know is present in human adults then. Okay, so this is going to occur in the cells of the kidney and also the cells of the liver. Okay, so in liver cells and also kidney cells. Now, we've seen something about liver and kidney cells before now. Remember, we saw that liver and kidney cells, they were those cells that had the type 1 5' 3' deiodinase enzyme, okay, and we will come back to that later on. So remember, they were converting T4 into T3, which was very important because T3 is a lot more potent than T4 at actually activating the thyroid receptor using that type 1 5' 3' deiodinase, and we'll see later on, I'll let the cat out of the bag now, that if you hugely restrict your caloric intake, so if you hugely reduce how much food you are actually consuming, so if you go on a very extreme diet, let's say the Atkins diet, a very extreme diet where you cut out all carbohydrate, or at least you reduce your carbohydrate consumption hugely, then the way that your basal metabolic rate actually goes down, I mean, everyone always talks about this, dietitians love this, so everyone always talks about the fact that if you go on a really extreme diet, it's counterproductive because your basal metabolic rate falls and therefore how much energy you're actually consuming goes down. The mechanism by which that occurs is not that T3 and T4 levels within the blood fall. That might be what you think might happen, but that's not what happens. Instead, what happens is the expression of type 1 5' 3' deiodinases in the liver cells and the kidney cells is reduced, and therefore their response to T3 and T4 goes down. Now, the reason that the hypothalamic-Pituitary-Thyroid axis is not overactivated by that, as we'll understand later on, is that the 5' 3' deiodinase that is in the hypothalamus and the pituitary gland is the type 2 one, which isn't affected by caloric intake. So they still see a normal amount of thyroid hormone in the bloodstream because they're still being stimulated by the thyroid hormones normally, whereas the liver and kidney cells don't get stimulated normally. But I will go over that again later on once we've had the full discussion of the hypothalamic-Pituitary-Thyroid axis. Okay, let's go back to how liver cells and kidney cells then are going to increase their, or have a heat production maintained by T3 and T4. So how do these cells produce heat? Well, they produce heat through what is known as a futile cycle, and indeed this is how a lot of heat production is done in the human body. Indeed, in brown adipocytes it's going to be a futile cycle as well, but in liver and kidney cells the futile cycle is slightly different to the futile cycle that will be in brown adipocytes. So what they're going to do is they're going to continuously do a process that requires energy. In this case it's going to be active transport. But then the active transport isn't going to be achieving anything because you're just going to let the ions move back again. Okay, so let me explain what's going to happen. So this is going to use the equilibrium or resting membrane potential across these cells. So I just need to remind you of how basic electrophysiology across cell membranes. So when you studied the way that neurons work for the first time long ago in physiology class, you will have learnt that across the cell membrane there is this equilibrium that is occurring all the time at the resting membrane potential between the movement of sodium into the cell and the movement of potassium out of the cell. So, let me just remind you. So, remember there are sodium gradients and potassium gradients across the cell membrane. So, sodium concentration in the extracellular fluid is generally around 145 millimolar. Okay, so this is sodium concentration. Sodium concentration inside the cell is generally around 12 millimolar. Potassium concentrations, meanwhile, you generally have a concentration of around 4 millimolar extracellularly and a concentration of 155 millimolar intracellularly. Okay, so you have a 12-fold concentration gradient of sodium favouring its movement into the cell. Meanwhile, you have approximately a 40-fold gradient of potassium favouring its movement out of the cell. Right. Now, what you're going to have in the cell membrane is leaky sodium and leaky potassium channels. Channels in the membrane that are going to continuously allow sodium ions and potassium ions to move through them. These are not voltage-gated channels, these are channels that are open all the time. Okay, so you'll have sodium channels and potassium channels in the cell membrane, and remember these are really important because the ions can't just move across the cell membrane without an ion channel because they can't get across the lipid bilayer. Okay, so let's say this is a sodium channel, so we'll have loads of these in the cell membrane. I'm only going to draw one, but remember, you'd have them all over the place in the cell membrane. And you'd have lots of potassium channels also in the cell membrane here. Okay, like so. Right, the other important thing to remember is that the permeability to potassium is usually slightly greater than the permeability to sodium. Okay, so usually the permeability of the cell to sodium is lower than the permeability of the cell to potassium. Now, what does that actually mean? That means that you're going to have more potassium channels than sodium channels. If you have more potassium channels in the cell membrane, then you're going to end up with a greater permeability for potassium than sodium. Okay, the ease with which a potassium ion will move across the cell membrane is going to be easier than sodium. So remember, you've got slightly more of these, and these might also be slightly more conductive to potassium ions. The ease with which a potassium ion can actually move across the cell membrane through one of these channels is going to be easier than for the sodium. Okay, right. So permeability to potassium is usually higher than permeability to sodium. Now, this means that initially, if you had no electrical potential difference across the cell membrane, you would get movement of sodium into the cell, okay? And you'd also get movement of potassium out of the cell because of the concentration gradients. So imagine initially that there's no electrical potential difference across the cell membrane. Initially, what would happen is you'd get a movement of sodium into the cell and a movement of potassium out of the cell just because of the concentration gradients. Now, because of what I've said about the permeability and also because of the difference in the scope of the concentration gradients, which of these is going to be the greater movement? Well, of course it's going to be the potassium movement. So the number of potassium ions moving out of the cell is going to beat the number of sodium ions moving into the cell. So you've got a net movement of positively charged ions out of the cell. Okay, now that's initially if you're running this from the start where you have no electrical potential difference across the cell membrane. But of course what this means is that we're going to have a net movement of positive charge out of the cell, and that's going to raise the electrical potential extracellularly and lower the electrical potential intracellularly, and that will create an electrical potential difference across the cell membrane. Okay, a voltage across the cell membrane. Now, as you create a bigger and bigger voltage across the cell membrane, which will be negative because you're lowering the electrical potential here and raising the electrical potential here, so when you move from here to here, you're going to be stepping down in electrical potential, that's going to oppose the movement of positive charge out of the cell. okay? Because positive charge wants to be where the electrical potential is lower, and now the electrical potential is lower inside the cell than outside the cell. So gradually, as this builds up and up and up, you're going to oppose the movement of positive charge out of the cell. That's going to be reducing the amount of potassium ions that are moving out of the cell and increasing the number of sodium ions moving into the cell. So as this electrical gradient builds across the cell membrane, you increase the sodium current in, decrease the potassium current out, until these two arrows are going to become equal to one another, and that's the point where you're at equilibrium, and that occurs when you get to around negative 65 millivolts as the electrical potential difference across the cell membrane. So this is the normal resting state then for the cell membrane. You have an electrical potential difference across the cell membrane of negative 65 millivolts, and at this resting electrical potential difference, the amount of sodium ions that are moving in from the outside of the cell into the inside of the cell through leaky sodium channels equals the number of potassium ions that are moving out of the cell through leaky potassium channels. Okay, so these two currents are balanced, and therefore there's going to be no net change in the electrical potential difference across the cell membrane. So that's when you reach the equilibrium electrical potential difference across the cell membrane, when the movement of sodium into the cell through leaky sodium channels is equal to the movement of potassium out of the cell. So at this electrical potential difference across the cell membrane, this current and this current are equal to one another, so that overall there's no net current across the cell membrane, and therefore you don't get a change to the electrical potential difference across the cell membrane. Now, of course, there's a problem with that, because this isn't an equilibrium ionically, this is an equilibrium electrically. You're moving the same amount of positive charge in that you're moving out, but it's not an equilibrium ionically. You're moving sodium ions overall in and potassium ions overall out. So to cope with that, what you have on the cell membrane is a pump which is going to exchange sodium ions and potassium ions, and this pump is, of course, the sodium-potassium ATPase. Okay, so the sodium-potassium ATPase will throw 3 sodium ions out and 2 potassium ions in, okay, and therefore it's moving the sodium back out, which has come in, okay, like so, and it's moving the potassium ions back in. Now, I know that the numbers that it's moving don't add up here, and it is electrogenic. However, trying to that into your understanding of the equilibrium just makes it far too complicated. So just imagine, understand this, that the main role of the sodium-potassium ATPase is not in building up the electrical potential difference across the cell membrane. Its role is just in reversing the ionic movements that there are at this equilibrium resting potential. Okay, imagine that it's not electrogenic. That's the easiest way to understand its actual function here. The other thing that you have to be aware of is there's also pumps that just move potassium ions into the cell, and they can sort of help balance the electrogenicity of the sodium-potassium ATPase. Okay, so if you imagine having another pump here that just moves a potassium ion into the cell here, okay, then that would sort of help to balance the electrogenicity of the sodium-potassium ATPase, okay, so that its effect on the electrical potential difference across the cell membrane is kept negligible. Okay, and of course these pumps will be using ATP to do this, so this will be hydrolyzing ATP down to ADP and inorganic phosphate. Okay, so that then is how we maintain the ionic concentrations on the intracellular and extracellular compartments. Okay, so to summarize then, the normal ionic equilibriums and the equilibrium of the resting electrical potential difference across the cell membrane, normally the resting membrane electrical potential difference is negative 65 millivolts, and remember, this is the electrical potential difference across the cell membrane that you need to have for the amount of sodium that is coming in due to its concentration gradient here across the cell membrane to equal the amount of potassium that will be moving out due to this concentration gradient across the cell membrane, and therefore you're going to get no overall movement of positive charge in either direction, and therefore it will be an equilibrium of the electrical potential difference across the cell membrane, because we've got no net movement of charge, so we're not going to disturb the electrical potential difference across the cell membrane. Now, however, there is the problem with having a net ionic movement. We've got a net sodium movement in and we've got net potassium movement out. The sodium-potassium ATPase, its role, and this is really important that you understand this, this is something that your physiology lecturers will have drilled into at the time. The role of the sodium-potassium ATPase is just in coping with this problem with the ionic movements. It moves sodium back out to counter this movement of sodium in, and it moves potassium back in to counter this movement of potassium out. Okay, and it uses ATP to do that. Ignore the fact that there is no, that it's unbalanced, that it moves 3 sodiums out and 2 potassiums in. The role of this is not to change the electrical potential difference across the cell membrane. This is built up by this, not by this. Okay, and that's a common misunderstanding, that this is the thing that's building up the electrical potential difference. It's not. Okay, and one explanation for how this has a negligible effect on the electrical potential difference across the cell membrane is that there are also active transporters which use ATP that just pump a potassium ion back into the cell, and they're going to help balance the number of potassiums coming in to the number of sodiums that are going out. Okay, so this doesn't really have any substantial effect on the electrical potential difference across the cell membrane. Its role, as you should understand it, is simply to maintain the ionic concentrations, because if we didn't have this, then we'd gradually lose our ionic concentrations across the cell membrane. Okay, right, so that's the normal equilibrium then that is occurring across the cell membrane. Now, how can we use this to increase heat production? Well, can you imagine what would happen if we increased the number of leaky channels in the cell membrane and increased the number of sodium-potassium ATPases in the cell membrane? Well, the idea is that if you were to, let's say, double all the numbers of the ion channels here, so double the number of sodium ion channels, so I'll draw another sodium ion channel here, and double the number of potassium ion channels. Okay, imagine doing this. So here are some more sodium channels, and here are some more potassium channels. So our hepatocyte, or our renal cell, has now doubled the number of sodium channels and potassium channels in its cell membrane. What's that going to mean? Well, that's going to mean that at the equilibrium, the overall number of sodium ions that you've got coming into the cell is going to be twice the amount it was previously. So if you double the number of sodium channels, the amount of sodium ions that are coming into the cell in a minute, let's say, is going to have doubled. Makes perfect sense? Good. Okay, same for potassium. If you double the number of potassium channels, then at the equilibrium, the number of potassium ions that you'll actually get going out of the cell due to the concentration gradient is going to be doubled. Now that will mean that the electrical potential difference will remain exactly the same, because we've doubled the sodium current in and we've doubled the potassium current out, so everything's still in equilibrium, so it doesn't change the electrical potential difference across the cell membrane. But now we have double as much sodium coming into the cell as we have, and we have double the amount of potassium going out of the cell. So we're also going to need to double the activity of the sodium-potassium ATPases. So we'll have to put in more sodium-potassium ATPases, like so, in order to maintain the concentration gradient, because we've got overall twice as much sodium coming in and twice as much potassium going out. Okay? And we'll still, if we do all of that, we'll still have the exact same equilibrium. Everything will remain in equilibrium, but now what's the difference? Because we've got twice as many sodium-potassium ATPases, we're going to be using twice as much ATP. So to maintain this equilibrium now, we've got the exact same equilibrium of concentrations across the cell membrane and the exact same electrical potential difference across the cell membrane, but because we've got twice as many channels and now twice as many sodium-potassium ATPases, we're going to be using twice as much ATP. So this is now going to be burning through ATP much faster, and where's all that energy actually going to go? Well, it's going to go into heat. This is the idea of the futile cycle that's going to be set up in liver and kidney cells, that we can double the number of ion channels in the cell membrane, double the number of sodium-potassium ATPases in the cell membrane. We'll still have the exact same equilibrium, but now we've got sodium, twice as much sodium coming in, twice as much potassium going out, and twice as much of it having to therefore be exchanged by the sodium-potassium ATPases. This is the futile cycle. The sodium goes in, is pumped out, the potassium comes out, it's then pumped back in. This is what we're referring to when we say a futile cycle. And where's all the energy that's being used to do this going? Well, it will go into heat. So, in liver and kidney cells then, a way that we can cause them to generate more heat is to get them to increase the number of sodium potassium ATPases in their cell membrane, and also increase the number of ion channels in their cell membrane. And this is indeed what T3 and T4 do in liver and kidney cells. Now, all that we actually know they do from experimental observation is we know that T3 and T4 going into liver and kidney cells will cause the number of sodium potassium ATPases in the cell membrane to go up. We haven't yet actually shown that the number of leaky sodium channels and the number of leaky potassium channels goes up. However, we know that that must happen, because if it didn't happen and you did increase the number of sodium potassium ATPases, there would be problems with the equilibrium, and the equilibrium isn't actually being disturbed. So we know that what must be happening is that leaky sodium and potassium channels must also have their expression increased by T3 and T4, but we haven't yet experimentally found which sodium channels and which leaky potassium channels are actually having their expression increased. But we know that that must, well, we believe that that must be what's happening. Okay, so I hope you understand what's happening here, that we are maintaining the same equilibrium, but we're using twice as much energy to maintain the same equilibrium by doubling the permeability of the membrane to sodium and potassium, and therefore having to double the number of sodium potassium ATPases in the cell membrane. And all of the energy that we're using to do that will now be being used to produce heat. Okay, right. So that's a mechanism for how T3 and T4 can tonically stimulate hepatocytes and kidney cells to be producing bodily heat. We'll have a break here, and in the next video I will talk about the effects of T3 and T4 on brown adipose tissue in the human neonate, which is also important for heat production.